part of the TCAP project, he's overseeing some student research there at his institution about beneficial and destructive nematodes of wheat and barley. And he's collaborating with Aaron Carter at WSU on that project, and it's been very successful. He's done a great job attracting students to participate in the TCAP research. So we're really happy to have Dr. Matute here with us today to tell us about nematodes. Martin, are you ready to go?
Yes, I am. Thank you very much, Dr. Sheman, for that, I don't say flattery, but for the very good introduction. I'll start first by thanking Amy yesterday, and she helped me. We put everything together, and she took me through everything that I needed to know. And she made it so simple, it looks as if anybody could actually do it. So thank you, Amy, for that great job you did yesterday. Our presentation this afternoon will be not so much as to what we've done research-wise, because we are just beginning, but rather we'll be kind of highlighting some kind of an awareness of plant parasitic nematodes, and also perhaps free-living nematodes. Not much of that today, but nematodes generally, as it relates to the TCAP project. So the main purpose of today's talk is to raise awareness to that, and then also at the end of the day we'll end up also probably let the audience know what is it that we do at the UAPB. So without much ado, we're going to go straight into what the talk is all about for today. We'll start with an overview of nematodes. Now, nematodes are the most abundant metazoans on planet Earth. They are the most abundant metazoans on planet Earth. And 4 out of every 5 multicellular animals in the world are actually nematodes. Now, for the most part, nematodes are cylindrical roundworms. They are mostly microscopic, especially those that are plant parasitic, and they are found just about everywhere where they can get food to eat. Now, they could be found in very pristine environments to extremely polluted environments. Now, this attribute of theirs, this is what makes it possible for us to be able to use them as indicators of environmental conditions. As Jamie said earlier, part of what we are doing in the TCAP will be also looking at the beneficial aspects of nematodes. Now, just to continue buttressing how widespread nematodes are, you could find them in very, very cold regions in the Arctics. You could find them in hot springs, you could find them on mountain tops, you could find them deep in the sea, fresh water, just about anywhere. If you actually went somewhere and nematodes were actually not found there, then it's going to be likely that that environment is extremely hostile to living organisms. Now, for plant parasitic nematodes, as I said earlier, they are exclusively microscopic. Now, with a trained eye, there are some female forms that can actually be seen, or there's some nematodes that can be seen with the naked eye if your eyes are actually trained, but you're not able to see any details. So that's why here I say that they are microscopic. Now, one way to distinguish between a plant parasitic nematode and a non-plant parasitic nematode is that plant parasitic nematodes, they have a stylet. A stylet is an anatomical structure they have around their mouth area, which is actually shaped like a spear. They use that for feeding. That's what they use in piercing plant material. Now, for root nematodes, There are three groups of root nematodes. We have the endoparasitic, we have the semi-endoparasitic, and then we have the ectoparasitic. Then we also have nematodes of stems, leaves, and seeds. So we can actually see from here that there do not seem to be any part of a plant that is spared of nematode parasitism. So nematodes could be a problem in any part of the tree. It just depends on which plant you are actually looking at, or which crop you're actually looking at. Now this is an example. This is an example of an endoparasitic nematodes. Endoparasitic nematodes, they are generally small nematodes because they have to live within the tissues. They have to live within the tissues of their plant host. Here you can see this is, I hope I'm not covering that. This is the stylet here. Remember we said it was more like a spear. This is the stylet right over here. And this roundish part you see at the base here, that's the stylet knob. It actually supports the stylet. So if this plant parasite, if it is feeding, this spear or stylet is going to be extruded. It's going to be extruded out of the animal so that it can actually sap. You can actually suck out plant sap. So this Pratylencos is a small nematode, and that's why you can actually afford to be exclusively endoparasitic. Then this, okay, this is still that's still Pratylencos. The next one, this is Hoplolymos. Hoplolymos is semi-endoparasitic. If you can see, generally it is a bigger nematode. It's a bigger nematode than the Pratylencos we saw earlier. If you look at the stylet here, it's supposed to be a longer stylet. And then this is a vulva here, so this tells us this is a female we're actually looking at here. This is the vulva right here. Now, semi-endoparasitic nematodes, about 20% of their anterior part of the body is embedded into the plant tissue, while the rest of the 80% is actually hanging outside. They are too big for the whole nematode to be contained within the tissues of their host plant. If that happens, the plant may die almost immediately. Then this is an example of an ectoparasite. And you can see here that what I've done is I have just half of the diagram here. These are extremely large nematodes, and you can see the stylet begins from here and then it continues right down here. Now, of course, it is understandable why the stylet is that long, because they are ectoparasite. They must be able to actually reach the root tissues. So all of these parasites, most of this stylet here is extruded outside the animal towards the lip region here, so they can actually make contact with root tissues and then absorb the nutrients that it actually needs to absorb. Shapes of plant nematodes. There are generally three shapes of plant nematodes. You have the cylindroid or cylindrical, we'll have the pear-shaped, and then we'll have the kidney-shaped. Now, this is an example of, okay, I hope I did not skip the first one. Okay, so this is an example of kidney-shaped or bean-shaped nematode. We have you can see the name here, Rotilenculus. So one of the things you're going to see here is it is only the female that is kidney shaped. This is the male over here, which is cylindrical. So this type of a situation will be referred to as sexual dimorphism. And of course, the reason why the females actually have this type of anatomical architectural design is just so that they have enough space to produce as many eggs as possible. Now with the male here, if you can see this structure, over here. Here they are, the two of them. They are called spicules. This is what the male actually uses to attach the female to itself so that sperm cells can be transferred from the male to the female. For this female here, we can actually see the vulva around this area. Now this is another, this one is pear-shaped. This is the root knot nematode, Meloidogyne, root knot nematode. The female is pear-shaped. This is the male, it's cylindrical or cylindroid. So again, this is another example of sexual dimorphism. So within the same species, the females, they look different from the way the males actually look like. Again, the anatomical design for the female here is just so that the female can actually contain as many eggs as possible. Now for this nematode, the one I showed previously, or root knot nematode in particular, they could have hundreds or sometimes close to about 1,000 eggs right in them that the female can actually have inside it. And from a parasitology or parasite perspective, having many eggs is a strategy for survival. Then now let's move to the economics of plant parasitic nematode. As I probably mentioned earlier, there's scarcely any plant which does not have its own share of plant parasitic nematodes. And this is very, very true, or more so for crop plants. Actually, plant nematodes, they actually compete with growers in terms of what they can get from these selected crop plants. And the plant nematodes are the #1 biotic cause of crop loss the world over. Now, the only factor that is responsible for more crop loss per year than plant parasitic nematode is drought. Drought only. So biotically, plant parasitic nematodes, they are actually the #1 cause of crop loss the world over. And the statistics I have here, I'm talking about up to 10% of total crop loss worldwide. These are statistics of up to 1989. And these statistics are actually based on in countries where they already know they have an awareness of plant parasitic nematodes, and where they actually try to take control measures. So in countries or in regions where they are not aware that plant parasitic nematodes are there and where no control measures are actually implemented, these values should be far, far higher. So, but if we deal with these values that we actually have, that I have presented here, now up to 10% of total crop loss is lost every year due to the activities of plant parasitic nematodes. And this translates to roughly 100 billion US dollars annually. That's a lot of money. And in cases where the nematodes are known to exist, up to $225 million are spent annually for nematicides. Nematicides are the chemicals that are used to control nematodes. So what makes plant parasitic nematodes so destructive? What is it that they actually have? What's their capability? So the first point is these are parasites in their own respect and pathogens in their own respect. I mean, as we all know, parasites and pathogens, they are destructive to their host. And if their host, because they get nutrients from their host. So if their host, in the case of parasite, they get nutrients from their host, and then in terms of a pathogen, it is what is that they actually produce that may actually end up making the host either sick or with less vigor. So nematodes are first and foremost, they are parasites and pathogens. Then we do also have nematodes that are active vectors of viruses. This is especially true for ectoparasites. I showed you, I showed the slide earlier on when I showed an ectoparasite, Zephynema. Zephynema, there, as we say, the ectoparasites, they do not live inside the root system. So they feed, they move from one plant to another. So if they fed on the plant that actually had plant viruses, as they feed in the next plant, which was a clean plant, they will go ahead and actually transfer those viruses into that next plant. So even if the nematode is not a problem to that particular crop, it has succeeded in injecting viruses in that crop that will end up being a problem. Then the next thing is nematodes are their enablers. In the soil, we do have bacteria and fungi that are pathogenic to plants, but they do not have a way of gaining entrance into the plants. So what happens is, once as a nematode feeds in a plant, either in the form of whether the nematode is an ectoparasite, semi-endoparasite, or an endoparasite, once that they feed into plant, or once that they get entrance into a plant, they create cavities or entry points. These opportunistic pathogenic bacteria and fungi, they will now get into the plant via these cavities or entry points that were created by the nematodes. Now when this happens, so you may actually end up having a situation where in the root system of a particular plant, you may end up having, you have a nematode there, you have viruses that were actually introduced, then you have bacteria, and then you have fungi. Those type of situations, they lead to what we call disease combinations or disease complexes. And cases of disease combination or disease complexes, they are more detrimental to the plant than if it was just one of these pathogens. Now, most of what I've discussed, I've said so far, these are not readily noticed. They are not readily noticed. Now, for the fact that they are not readily noticed, it does not mean they do not exist. In areas where it is where an awareness of nematodes already exists, you may have nematologists going out into field plots and collecting samples. They may collect samples year 1, year 2, year 3, right up to year number 19, and they may not ascertain that there is a nematode problem. But for the fact that up to year number 19, a nematode problem was not detected, it does not mean that a nematode problem may not arise by year number 20. So the reason why I'm saying this is, even if we do not have a nematode problem this year, it is just a matter of time. The only way that we can actually stem the activities of plant parasitic nematodes is that nematologists, they have to be doing the monitoring and all the sampling every year or every time, so that once as a problem begins to arise, they will know that it is there, and then corrective measures will be taken. Now, so how will you recognize that a particular field or a particular plant is actually infected with plant parasitic nematodes? Now, as you can see here, I have a couple of above ground symptoms here. Now, I must say very quickly that these symptoms are not only limited to nematodes. Other pathogens, I know if you have any plant pathologists among us here, they'll attest to that. Other pathogens can also bring about these symptoms: dead plants, stunting, chlorosis. The only way, the reason why these are actually we know for certain that these are ascribed to plant parasitic nematodes is because, number one, control experiments have been carried out, and then number two, if it is a field situation, one has to go out there physically, collect the samples, either the root samples and the soil samples, and then go back to the lab and actually make the analysis. However, these are some of the above ground symptoms: dead plants, stunting, chlorosis, yellowing of foliage, reduced yield, loss of crop. It could be slight to total. It could be slight to total. So these are some of the These are some of the above ground symptoms of nematode infection. And this is a wheat field that has been damaged by nematode. You can see here that this is another field over here, but this is not wheat. You can actually see here, you can actually see over here where I actually have the pointer that these are kind of say patchy spots in the field. These are patchy spots in the field. The other areas further up there that you have a large level of plant vigor, it will certainly going to have little or no nematodes, but these areas, these patchy areas, if you did any sampling here, you're actually going to get a lot of nematodes. You can see that the plants there, they are barely surviving. Now this is another example over here where we have yellowing or chlorotic leaves. And of course, we know once that leaves become chlorotic, then or yellowish, then sometimes their ability to carry out photosynthesis becomes simply seriously compromised. This is another patchy field here where nematodes actually abound. Again, you can actually compare that with other parts of the field where you have luxuriant growth. So what are some of the below ground symptoms of nematode infection? Again, like I probably said earlier, it may be difficult to distinguish that from other biotic and abiotic factors in the soil. So the best way to know is that a diagnosis will have to be done. If it is the roots, you take the roots and you see if you can extract any nematodes from the roots, or if you can extract nematodes from the rhizospheric soil, that is soil around the rooting system. Now, but again, with a trained eye, if you took a root system that was infested with cyst nematodes, they are relatively large nematodes, and that you should be able to actually see with the naked eye, if with a trained eye. Now, other symptoms that you're going to see with the rooting system is going to be the reduction in size, pruning, lesions, stubby, root swelling or root knot, root proliferation, coiling, and so on. Now, if all of this happens to a root system, the affected root, the root is going to be affected anatomically and physiologically. And once the root is affected anatomically and physiologically, then the function that it's actually supposed to carry out, for example, anchorage or the absorption of water and minerals from the soil, all of that actually is going to be compromised. And if that is compromised, then of course we do not expect the plant or the crops to actually do well in the field. Now, so this is Heterodera avene here. It's a nematode that has been reported to be very, very devastating in wheat. This whitish roundy stuff you actually see in here, these are the females. These are the females. So like I said, with a trained eye, one can actually see this even just using a hand lens, and you know that okay, there's actually a nematode problem there. Then here you can actually see this is a root system that is actually being infected by nematodes. You can actually see that it does not look like a normal root system, and therefore it cannot perform the job of a normal root system. Now this is another one I put here, but this is not wheat. This is for tomato plants. This is for tomato plants. I put it here just for demonstrative purposes. Now on here, this is a root system that is infected with the root knot nematodes. So you can see the root system here has a lot of swellings or knots like the knot in your tie. While on my right here, this is the normal root system. This is the normal root system on my right here. Of course, while the rooting system on my right is going to perform the normal function of the rooting system, the one on my left will scarcely be able to be functional. Now this is just some more examples of a rooting system that is infected with plant parasitic nematode. The swellings we see here, they're actually caused by the root knot nematode. So if you took any of these swellings and you macerated them, you should be able to find all of those nematodes in there, and with all of their developmental stages from eggs to the adults. after giving that introduction, so the next question is going to be, so what is the let's say something about the triticale and the nematodes. Now, as we are aware, the two crops that the TCAP project is actually working with is barley and wheat, and based on a known statistic, but this was about since 1989. 6.3% of all barley that is actually produced is lost annually due to the activities of plant parasitic nematodes, and this is estimated to be more than $1.1 billion in loss every year. For wheat, it's even higher, 7.0%, and this is estimated at about more than $5.8 billion US annually. I think there's a lot of money and a lot of time wasted in terms of the growers. So with these figures, even if they are not current, one may want to ask the question: the triticale nematodes, are they important? I'm sure the answer is going to be yes, that they are actually important. So what is our role in the TCAP project? What is our role in the TCAP project? As Jamie said earlier, our group, we have two main functions. We have two main functions for the TCAP project. Number one is, as new lines of these crops are actually being bred or developed, we will actually be screening them for nematode host status or nematode resistance. We'll see whether they are resistant or very susceptible to plant parasitic nematodes. Our second job is we'll look at the free living nematodes in the soil. We'll look at the nematode soil, the soil nematode food web structure. Using the free living nematodes, we have the bacterial feeding nematodes, the fungal feeding nematodes. Then we'll have the omnivorous nematodes, and we'll have the carnivorous nematodes. Now, using these four trophic groups depends on the types and the number that we actually find in the soil. That could actually be weaved into their into a structure, into a nematode food web structure, which will be able to tell us whether that soil is healthy or not healthy. We can also use that information to monitor whether the soil conditions are changing or not. As I was discussing with my colleague Aaron Carter and WSU, we're saying that, well, in as much as we want to grow lines of wheat or barley that are resistant to pathogens or high yielding and all of that, we also want to be sure that the crops that we're actually growing, we also want to exactly know what impact do they have on the environment. So again, that we can actually use nematodes in doing that. Jamie also mentioned this earlier. We collaborate with Dr. Aaron Carter, Washington State University, in the Crop and Soil Science program. Now, our study fields, we have them in Washington State and we have them in Arkansas. The three we have in Washington State, McGregor is for spring wheat, and the other two are for winter wheat. Almoto and Colton, they are for winter wheat. In Arkansas, only winter wheat is actually grown, and these are our three fields in Arkansas: McDonald, Turner, and Algoa. Now, the way we carry out our field studies, we'll be collecting samples in our fields 3 times per growing season. We're going to collect samples pre-plant, mid-season, and harvest. Now, for our fields in Washington State, we have actually completed our initial soil sampling for all of the fields. We are now actually in the next couple of weeks, which will be doing our mid-season sampling. Doing our mid-season sampling in the next couple of weeks. In Arkansas, the pre-plant sampling, that's towards winter, is actually in progress. But just to give you an idea, if we let's take one of our fields in Washington State, Almoto, where this is what these are the 4 species of plant parasitic nematodes that we've actually found in the group that we call the CP2 or PL2 group, colonizer persister group. We'll be able to we identified for our initial profile, we identified 4 genera of nematodes in this CP2 at the CP2 level or CP2 group, and then this is the one. If I go back here, now among them, this one here, the Silencus hilarurus, this was the one that we actually found. This was one that we found in all of the samples. Now, these are nematodes of the CP2 group or PL2 group. They are generally very small nematodes. So you can actually see this is a very small nematode. And nematodes that are very small in size, not that they are not economically important, they have to be in very large, very large numbers. So they feed on plant bed by virtue of their size. Their destruction is not as great as larger nematodes. Then the other class of nematodes we found in our pre-plant soil profiling of Almoto in Washington State. We had 4 genera of 4 species of nematodes. We have the Syriasis nematode, lance nematode, the root knot nematode, and the stunt nematode. Now, among these 4, the one that was dominant was Heterodera avene. That was one that was actually dominant among these 4. Now, this is the Syriasis nematode here, Heterodera avene. This is the female. Now, this stage in the middle here, it looks like sausage or banana. It is destined to be a female. So Heterodera is one of those type of nematodes, as we said earlier, that it practices sexual dimorphism. So the larval stages, after larval stage 2, if they're going to be females, then they'll start bloating out, as we are seeing here. So it means that this is a larval stage 3, which will end up actually being a female. Then of course this is actually destined to be a male or a male already. And then this is a wheat root here, and you can actually see these are the nematodes right here. You can actually, again, with a trained eye, even if you do not use a microscope, you may be able to see something that will tell you that, okay, I think those are the nematodes I'm actually looking at. Now here, this is a Heterodera female. Now this yellowish part we are seeing here, this is an egg mass or an egg sac. So the eggs, once they are produced in the female, this is where they actually mature. They mature here. They mature here, and then they will be released to the. Later on, this egg sac is actually going to explode, and all of the eggs that it contains, sometimes hundreds or thousands of eggs, they are going to be released out into the environment, and then they will hatch, and then the cycle will actually continue. Now, so based on our initial sampling that we've done so far, we've realized that among the plant parasitic nematodes, now Heterodera avene and Silencus hilarurus, these 2 genera, two species were actually recovered from all the samples. Now, the Heterodera, because it is a CP3 level nematode, it is more destructive than a CP2 level nematode. And from our initial sampling thus far, we found out that up to 272.2 were recorded per 100 milliliters of sample of soil. Now, from every standard, these are actually high initial population. These are actually high initial population. Now, because of this, we don't know though, but because of this, we are actually expecting that if any of the new breeds that my collaborators actually working with. If any of the new breeds if any of the new breeds we planted in this field, if any of them were planted in this field that is susceptible to plant parasitic nematodes, then we are actually expecting that Heterodera avene may actually end up being a problem. However, we'll have to wait and see whether it's going to be true or not. If we end up realizing that some of the lines that are actually bred there, they end up developing high populations of Heterodera, then that's good for us. If it ends up being that they are very, very susceptible to Heterodera, then that will not be very good news for us. These are the students that are in our lab, and they've been working very, very hard, and they are actually looking forward to some internships this summer in Washington State University. All of that was actually organized by my collaborator, Dr. Aaron Carter, and also Dr. Jamie Sherman. So where, as Jamie said earlier on, we are at the Pine Bluff. We are actually taking our investigation of nematodes of wheat very seriously, and we are hoping that other teams that are associated with the TCAP project, that they will also want to start thinking that nematodes are very, very important when it comes to crop production generally. Thank you.
So it looks like we have some time for some questions. Yes, Tyson's applauding. Thank you so much, Dr. Matute. But I'm wondering if anyone has any questions. If you have a question, you'll need to chat in typing into the chat box, and we'll read the questions, and then Martin will answer them. So any questions from the group? Dina's typing in. So hold on a second and we'll see what she has to say. She says she doesn't want to ask a dumb question.
No, there's no dumb question. There could be a dumb answer, but not a dumb question.
Is there plant host resistance to nematodes? Is her question.
Right. Yes. Oftentimes what happens is plant breeders, they have come on to, they develop some crop varieties or cultivars that are resistant to nematode. For the first year, that resistance holds, but by the time the crop is planted the second year, that resistance is broken. So I am not aware that we actually have crops that have successfully been grown for nematode resistance. What some plant breeders do now somewhere in California that I know is for something like grapes, they actually take one type of a plant and they kind of superimpose it on another plant. I don't know what I don't know what terminology is used for that. But as if they have something like a rootstock that they put in the soil, and then then the plant itself, say the grape plant itself, is kind of say attached to or grafted to that rootstock, and then it grows without nematode problems. But oftentimes the plants that have been bred for nematode resistance, after one or two crops in the field, that resistance is broken, the nematode problem continues.
Yeah, that's really interesting. So, Dina, I guess you answered her question, and she says thanks. Amy asks, grafting is the horticulture term. Oh, she's letting
okay,
you.
grafting.
Yeah.
Okay,
Yeah.
all right. Yes, I guess that is yes. Thank you.
So I guess that's a function of how many different types of nematodes there are that the resistance gets overcome. And Dina said, I guess that's job security for plant breeders. It sounds like an area that needs a lot of work.
I agree.
So I'm glad you're drawing our attention to it. Tyson asks, if nematodes cause such large losses, why are more nematicides used, or why are they not? Why aren't they not? So how do the nematicides work, Martin, and what is some of the factors limiting their use?
Okay, well, I'm glad that Tyson is from UC Davis. Yeah, I actually did my postdoc there. But one of the problems that I don't know that Tyson knows Dr. Howard Ferris. I worked with him when I was out there. One of the problems that they actually found in California was the nematicides that we're using for controlling nematodes. They started finding this nematicide in groundwater. So the state kind of banned the use of nematicides. So for the most part now, the permits are rarely given for the use of nematicide. They are given sometimes, but they are rarely given. And so what nematologists try to use for the most part now is using non-chemical ways of trying to control these nematodes. It could be via crop rotation, where you use them. So if, let's say, you planted, let's say, wheat this season, and the dominant nematode was, say, Heterodera avene. Now, if we know that corn, for example, is not very susceptible to Heterodera avene, then we may want to add corn in that rotation, so that way, maybe by the time you bring back wheat into that rotation, the population of Heterodera must have reduced to a level where it will not constitute that much damage. So crop rotation, it's one of the major ways in which nematodes are currently being controlled or managed to a very large extent.
So it sounds like it's really important to know which nematodes infest which
Exactly.
plants, and then also to know what's hitting your
Yes.
crop, so that you could plant something different. Yeah, that's
Exactly.
interesting. So are the nematicides harmful to other
Yes.
animals? Is that why they're controlling
Yes.
their use?
Yes. So if you put nematicide in there, of course nematicide, they are chemicals. So when they go in there, they are not only killing the nematodes, but they'll end up probably also killing some of the bacteria or the fungi that actually end up contributing to soil fertility as well. So it ends up having they end up acting more like some kind of an ecocide. So it's not going to be specific in killing only nematodes, but it will actually end up killing other beneficial microorganisms in the soil. And even if it was killing only nematodes, as I said earlier, we do have beneficial nematodes. If for example, if you have an abundance of bacteria feeding nematodes or fungal feeding nematodes in the soil, that's very good for you, because with bacteria feeding nematodes, if when they feed on the bacteria, any nitrogen in excess of their body, they excrete that to the environment, and this nitrogen that the plants actually need. The same goes for the fungi. So we have beneficial nematodes in the soil, apart from actually using them as indicators of soil conditions. Free-living nematodes, if you have them in very large quantities in the soil, they are what we may call the decomposer, and by that they do a lot of mineralization in the soil, which is good for plant growth.
So are there ways to increase the number of beneficial nematodes in the
Yes,
soil?
one way I've been using organic organic sources of fertilization. Might be organic sources of fertilization.
So manures,
yes.
and etc.
Yes. So manure in particular will actually end up increasing the number of bacteria feeding nematodes, number of bacteria feeding nematodes. And then with that's for animal manure. Then if it is plant manure, that will probably have a tendency of maybe increasing more of the fungal feeding nematodes.
Oh, that's interesting. So the organic makeup of your soil affects which nematodes are present.
Yeah. Yes.
So how do you end up getting a really bad concentration of some of these pathogens? Is it by repeating the same crop over and over again? Is that a primary issue? Or?
Yes. So that's one is repeating the same crop over and over again. Number two, also it could be that you are planting if the stock that you are planting, if it's already infected. So it just depends on what you are planting. So in some cases you are planting material that is infected already. So once you put it in there, the nematodes just bloom. And let me just take one step back. Nematodes, they can complete their life cycle within between 3 weeks and 4 weeks. So the turnover for their reproduction, especially the smaller nematodes, is very, very high. Very, very high. Yeah, so if you plant the same crop in the same field for a long period of time, that's recipe for high population build up. If you planted already infected material, that will also be a problem. Sometimes irrigation water also, it depends on where you're getting your irrigation water from. If it's coming from a stream or from a river which actually have nematode, then what you'll be doing is that you just simply be piping nematodes into your field. So there are so many different ways, and if a nematode problem is actually suspected and a nematologist comes around, those are the different things that different questions will be asking and actually testing so as to actually know exactly where the problem is actually coming from.
So you seem kind of surprised at how high the rate was of nematodes in, I assume it was the WSU soil that you were looking at. What do you have an idea of why it was so high?
Well, I think some of those fields were already planted with wheat before. Some of those fields were already planted with wheat before. So I think that should be the only explanation for that particular one. So if let's say they had a wheat crop there 1 or 2 seasons back, and those nematodes, one thing with nematodes is that you cannot completely eliminate them. If they do not have proper sources of food, their population can be dwindling gradually, but they cannot be completely eliminated. So I'm suspecting that field already had a wheat crop either the year before, or maybe I have that somewhere in my notes somewhere, either the year before or 2 years back. I think that may be the only logical explanation. In other words, there was already a crop in that field that was very susceptible to heterodera.
Mm-hmm. So I'm sort of monopolizing. Does anybody else have a question? David May is typing, so we'll wait for him. One thing that you can think about while we're waiting for him is I'm wondering how long does it have to be between you, how long do your rotations have to be before you reduce your load enough? So say I planted wheat this year, how long do I need to wait before I could plant it again?
That question would depend on the load of nematodes that we actually have in the soil, and it's something that for every field or for every farm it will have to be studied. In some cases it could be after 1 crop you rotate. In some other cases it could be after 2 crops you rotate. And in some cases, it could be that if you planted, let's say, wheat this year, you have to take for about another, after 2 growing seasons or so before you actually come back there. That's when the population must have been reduced significantly. So in each of these situations, you actually need some kind of a specific location study. There really isn't any one type of treatment that you can say, okay, maybe after every one crop you actually rotate. No, some specific study has to be done in a particular field for you to come up with that information.
Interesting. Well, David is still typing, so we'll just wait. Oh, he wrote a lot. Was the fact that nematode resistance was overcome after one growing season due to subsequent infection with a separate species of nematode, or was it due to the fast generation time of plant parasitic nematodes and survival of resistant individuals?
All right, thanks. For the most part, if you go to any particular field where you have a particular crop species, you will find out that you end up having just one major species. You may have a secondary and a tertiary one. So if it is the same field for the same crop species, the problem is with that one particular species, and its population will keep on growing so long as it actually has a suitable host to actually feed with. So for the answer to his question is the same species. It's just that it grows faster because it has an abundance of food supply.
David, do you have a follow-up to that, or did that answer your question? So while David's typing, some of you may need to go. I just want to thank Dr. Matute again. David says, I'm a little confused because I thought that a resistant variety planted would actually reduce the reproduction rate of the nematodes.
Yes, there's no confusion about that. If you have a resistant variety there, that means that the nematodes do not have a suitable source of food, so their population is going to dwindle. But if I understood his first question, he was asking, I did not understand that was what he was asking for his first question. If I understood his first question, the question was the fact that nematode resistance was overcome after one growing season due to subsequent infection with a species of nematode, or was it due to fast generation time of plant parasitic nematode survival? Okay. Alright, okay. I see what he's saying. Yeah. If resistance is broke, yeah, okay, he's correct. Okay, I did not understand his question. This question he was asking is, if you planted a resistant variety and after a while it became, if the resistance is broken, is it broken for that nematode, or is it that a different nematode that's actually taking the niche of that previous nematode? The answer is correct. It becomes resistant to another species. Because it's not another species, that's correct. Or rather, it becomes right.
Okay, so what you're saying is that
to another species.
When it's overcome, I see. So when it's overcome, it's because a new species has come in that's also parasitic. Yes. Okay, got it. Okay, David, does that answer your question? He's typing, so. Yeah, so he's interested in winter wheat breeding for nematode
Yes.
resistance.
Yep.
And he's just started here in Montana, so he's just starting to work on this with the winter wheat breeder here in Montana. So he's particularly
this will be interesting since that he's specifically breeding for nematode resistance. This will be very interesting.
Yeah, and so they're just starting to, I think, to get an idea of what's present and all that sort of stuff. So maybe you guys could talk later. So Tyson asked, so would alternating between resistant and non-resistant varieties be as effective as rotating crops? Or maybe different sorts of resistance? Tyson, maybe that would work if you had a line resistant to this type of nematode, and then that's interesting. Probably no one's done it because we don't really have a crop maybe that we know, or we don't have a weed that we know is resistant.
Exactly. That's why I'm saying. So at the end of the day, all of that investigation has to be done. All that has to be actually be done, and then okay, this crop is a non-host, so it will be essentially rotating non-host and host crops.
Yeah, interesting. So Dina's saying, I think there are resistant soybean varieties.
Yes, some have actually been bred for, they've been bred for resistance. The resistance, as I said, it will hold for a while, and after a while it's broken up because other nematode species, they actually come in and they start infesting. That's why I said earlier that while they may not be a problem in the 19th year, they may end up being a problem in the 20th year. Yes.
Yeah, I see. And David says there are several good sources of resistance to Pretiolenca species. Yeah. So only to
species, yes.
Nematodes. Yeah, the certain. Yeah, David says in winter and spring. So there's other nematodes that can infest. I see. Well, our numbers have dwindled, but I'm sure that Martin would be willing to answer any questions. If anybody has a question, I can get his email to you. We really appreciate it, Martin. I really enjoyed and learned a lot today, and so thanks a lot.
Thank you all for listening. That'll be great. Yes. So David, I'm looking forward to him contacting me. I'm really excited that he is working specifically on nematode resistance. I'm really excited about that.
Great, and I'll get him your email, Martin. I'll make sure he has it.